In this module, we will discuss where messenger RNA it is localized in the cytoplasm. And this regulatory umbrella of cytoplasm and the messenger RNA, it includes, number one, localization of messenger RNA to certain sites within the cell. How we know where messenger RNA is localized? Then whether or not a messenger RNA is translated, and if so, how often it is translated. You see, because messenger RNA is to be translated into proteins, and the regulatory also is that the half-life of the messenger RNA, which is the property that determines for how long the messenger RNA can be translated or should be translated. So, if we see, there are three important parts in this. One is the localization of messenger RNA. Second is that whether or not messenger RNA is translated. And third is that the half-life of the messenger RNA, that is its life, which is very important for the regulatory process. Now, transitional level control, it operates through interaction between specific messenger RNAs and various proteins and micro RNAs which are present within the cytoplasm. So, if you imagine or if you see that in the cytoplasm, what is present? That microRNAs, proteins, and different microRNAs, messenger RNAs, they all are present in the cytoplasm. But where the messenger RNA is localized. Now, another important thing is that it is not essential that all microRNA, all messenger RNA is to be translated. It is just like this. If you read a sentence, it is not important that in an English sentence, all part of that sentence are important. There are some important portion and some are not. So it is discovered that in the messenger RNA, it contains certain non-coding segments as well. There are segments which do not code anything, and they are called untranslated regions, UTRs. And they are untranslated regions. They are at the both ends, 3' ends as well as the 5' ends. There is present UTRs. And these UTRs, one which are present at 5 end, we call them 5' UTR, and they extend from methyl guanosine cap at the start of the messenger to AUG initiation code. You know, these are the different codes, different areas, segments where the initiation occur. Whereas the 3' UTRs, they extend from the termination code at the end of the coding region to the end of the poly A tail that is attached to nearly all eukaryotic messenger RNA. So, untranslated region of 3', they extend from termination codon to the end of the codon, whereas the 5' untranslated they are from methyl guanosine cap to the start of the message to the AUG initiation. So initiation and the termination end. We can see in this diagram that there are untranslated regions at the 5' region going on there. Then there is a coding region in between. Then untranslated region is there, which is at the 3' end and extends up to poly A end. That is the untranslated region. They contain nucleotide segments used by cell to mediate transitional level control. Now, cytoplasmic localization of messenger RNAs, that is the localization of specific messenger RNAs to specific region of the cell cytoplasm. That is, in the cell, the specific region of the cytoplasm, the specific messenger RNA, they are to be present. And there is a major mechanism by which cell establish the functionality which is distinct cytoplasm domain. So in the cytoplasm, there are different domains and the different cytoplasmic areas, cytoplasm, messenger RNA, which are there. Now, the information that governs the cytoplasmic localization of a messenger RNA, that resides where? In the 3' untranslated region. So the information which governs the cytoplasmic localization, that where the cytoplasm will be localized of the messenger RNA, that reside on messenger RNA 3' untranslated region, just as shown in this diagram, that the cytoplasmic localization of messenger RNA in the fruit fly, where it is occurring at the different sites of this. And in this case, we will see that the example of the fruit fly development, and considering the fruit fly eggs, larvae, and adult stages, where the different position of the messenger RNA is there. Now, the development of the anterior and posterior, that is the head, abdominal axis of the larva of the fruit fly, and subsequent adult, that is foreshadowed by the localization of specific messenger RNAs along the same axis in the oocytes, which is shown in the figure. So there are the different positions, different places where at specific places where the messenger RNA is situated or is present in the cell.